Thanks a lot for the introduction, and thanks a lot for virtually having me there today. I'm really sorry I couldn't join in person, but I had already agreed to present at a different event. But I hope this still stimulates an interesting discussion there, and I look forward to joining perhaps the next time in person. So today, what I'm going to talk about, I think it's going to be a little bit tangential with respect to the main focus of this conference, because the work that we do, and in particular the work that I'm going to discuss in the next few minutes, is really more kind of biophysics compared to some of the talks I have seen today. And I'm going to talk about this idea that we have and many have around building devices that look and behave like biological cells, and we call them synthetic cells. And in particular, I'm going to show how you can actually do this using self-assembled DNA nanostructures and engineer reaction diffusion processes happening within these DNA-based synthetic cells in order to kind of create interesting architectures as well as interesting functionalities. This is a very brief outline of the talk. First, because I think this might be new perhaps to some of the audience, I'm going to spend just a few words introducing the concept of synthetic cells, what they are, why we are interested in them, and then I'm going to discuss how we can build them with DNA nanostructures and pattern them with reaction diffusion. So the idea of synthetic cell is not new, but is really becoming more and more popular, and the community is growing very rapidly. It's a very ambitious idea. So the idea is that we want to start basically from inanimate molecular components, which can be basically anything you can put your hands or pipettes on. They could be biological, so you could have enzymes, transcription and translation machinery, nucleic acids, or they could be inorganic, like nanoparticles or synthetic polymers. And then we put all of these together, and you can really use tools that come from a lot of different disciplines. There's people who approach this from a synthetic chemistry perspective, and there's others like us who approach this from a more biophysics perspective. And the idea is to try to build a device or an array of devices that kind of behave a little bit like biological cells and also look a little bit like biological cells, as we are going to see. And the kind of behaviors that we aim to implement in these synthetic cells, there can be many of them, depending on what exactly it is that you want to do with your synthetic cell. You can aim to sense and detect biomarkers. You can aim to engineer communication between with other synthetic cells or with biological cells. You can engineer these devices to produce things like nucleic acids or peptides. You can engineer them to move around, for example, following a concentration gradient, and even to grow, divide, and perhaps in the future also evolve. And there are many good reasons why one might be interested in kind of going through the hassle of designing something that, at least in principle, could be complex and very challenging to make. A very good reason for this is that you can use these devices to study emergent complexity and basically try to address biological problems. And a lot of work has been done by many in this direction. This is some work that in the group we've done for a few years using very simple synthetic cell models decorated with DNA nanostructures to look at multivalent interactions between membranes, which are of course relevant in a lot of different biological problems like cell adhesion, motion, mechanosensing. And we developed, using these precise experiments, a statistical mechanical model in order to describe these interactions. There are many in a slightly kind of parallel community looking at protocellular models, which are built in a way that is realistic with what people think would be the chemistry, would have been the chemistry in the early planet, in order to basically study the emergence of life and prebiotic evolution. And there are many applications, or at least foreseen applications, of synthetic cells for actual technologies. And there is growing interest from industry for healthcare applications of synthetic cells. The holy grail here would be that of building perhaps an immune synthetic cell that you could put in the body, and then it could sense the presence of a certain biomarker and then perhaps migrate towards the site of a certain disease, and there produce and release therapeutic compounds, so to have a kind of low toxicity therapy. This is, of course, something that is perhaps a couple of decades away, but there are more approachable applications, for example, to constructing synthetic blood, or slightly smarter drug or vaccine delivery systems, or diagnostic and therapeutic, or sensing and diagnostic platforms. So the way in which I like to frame the problem of constructing synthetic cells is that really you need to look at two kind of main aspects. So one of them is the structure, and the other one is the information processing. So for the structure, what you really need is to build a microcompartmentalized device which basically can separate the inside from the outside of your synthetic cell, regulate transport between the two, and also, importantly, create internal heterogeneity. So give you ways to kind of partition your synthetic cell into different compartments and localize different molecules in different bits of your synthetic cell. So you can spatially distribute functionalities, a little bit like it happens in biological cells with the organelles. You can achieve this using, for example, membrane systems that you can build from synthetic lipids or from synthetic polymers, for example. But more and more popular are becoming so-called membrane-less implementations of synthetic cells, which rely on things like synthetic condensates, as we're going to see, actually, in the research I'm going to discuss, or things like coacervates or hydrogels. And these you can still use to kind of regulate transport in and out of your synthetic cell. And as we're going to see, you can still use to engineer internal compartmentalization. Then you need a source of programmable molecular machinery that operates within your compartment and does the information processing, in air quotes, which really means everything from kind of sensing stimuli, transducing signals, communicating, storing information, perhaps replicating information. And typically you would do this using reconstituted transcription and translation machinery, or you can also use synthetic nanostructures that can sustain molecular reaction networks, as we are actually going to see in a second. So in my research group, we try to basically use the tools of DNA and RNA nanotechnology, combine them with kind of my original background in soft condensed matter and biophysics to try to contribute to this emerging field of synthetic cells. And we are, of course, not the only ones. There are many across the world actually looking at synthetic cells from a similar angle. These are just some amazing publications from some groups in Germany, in Japan, and the United States that you can look at to learn more about this, but there are many more around. So from kind of our little corner of England, we try to do our best to contribute to different aspects of synthetic cell engineering using DNA nanotechnology. And we have work looking at signaling and communication and interactions with biological cells, tissue formation, motion, and then the creation of these compartmentalized cell-like architectures and the spatial engineering of functionalities within them. And that's what I'm going to enter into further details now. So I'm going to show you how we can use the self-assembly of DNA nanostructures to build membrane-less type synthetic cells and arrange different functional components within them using reaction diffusion. Before we look at the reaction diffusion, we need to learn about the actual material, which is going to constitute the scaffold of our synthetic cells. So this is something that we have introduced a few years back, and it's a material formed by very simple DNA nanostructures, just about as simple as you can make them. These are fixed holiday junctions, or nanostars, as we call them, but they have one kind of peculiar characteristic, which is that we decorate the end of each little DNA arm with a cholesterol molecule, which makes these objects amphiphilic. The way that you process this to make them assemble is that you simply heat them up and then very slowly cool them down. The single strands will assemble into these four-way junctions, which will also assemble, forming an extended network due to the hydrophobic forces between the cholesterol molecules that will kind of all come together into little hydrophobic pockets, like micelles, which will be then cross-linked and coordinated by these four-way junctions. So if you cool this system slowly enough, you will actually form crystals. And you can see this very obviously if you look under the microscope. And you can also see this if you do X-ray scattering, as we're going to see in a second. If you cool this a little bit faster, or if you change design in ways that I don't have much time to discuss, you can control the viscosity of these objects. You can actually make them liquid, you can make them amorphous and gel-like, or you can make them still crystalline, but still having a round shape. We are going to focus primarily on these round objects. We're going to refer to them as DNA condensates. For the case of the crystals, you can figure out that they are body center cubic by doing small angle X-ray scattering at the synchrotron. You can measure the lattice parameter of these crystals, and actually you can control it very precisely by changing the length of your little DNA arm. And you can do so with single nucleotide precision. And you can tune the lattice parameter between 18 nanometers in this example, all the way to 35 nanometers. And this is very useful because by changing the lattice parameter, you're basically changing the pore size of these crystals. So you can make them more porous, so larger molecules can diffuse it freely, or less porous, so to block diffusive transport. And that's very useful, as we're going to see later. We can also functionalize these materials, for example, to make them capable of accumulating and releasing protein cargoes. This is easily done by attaching a nickel NTA modification halfway along these little DNA arms. And then you just introduce, for example, some his-tagged recombinant GFP that will be uptaken in the crystals. And then if you chelate away the nickel, the GFP will be released, and you can repeat this. So capture and release proteins. We can also engineer structural responses in these materials. The simplest thing you can do is to trigger their disassembly using Toehold mediated strand displacement. So you add oligo, which basically peels off a little bit of DNA that links the four-way junction to the cholesterol. So after the displacement, the cholesterol is no longer linked to the holiday junction, and the materials simply fall apart, releasing whatever you have encapsulated within them. You can also make these responsive to other stimuli, for example, the presence of certain cations, which we have done in collaboration with Marco Di Antonio, who is also in this conference. So I don't know if he's, yeah, you can see someone waving, so maybe he's there. So what we have done here is that we have replaced the central junction of these stars with a tetramolecular G-quadruplex, which only becomes stable in the presence of potassium. So all you do is that you add potassium, these stars will form, and then they will start assembling, and you form the condensates. And then if you chelate away the potassium using krypton, they will instantly disassemble. So this was a bit of a kind of quick overview on this material that we have worked on for many years, but it's just to give you a flavor of what we can do with this, and to convince you that this would really make interesting materials to act as scaffolds of synthetic cells, so membraneless synthetic cells. This is because we can make them the size of a cell, we can functionalize them, we can give them the ability to capture and release molecules. They are actually very stable, we can make them responsive to loads of stimuli, and we can regulate diffusive transport through them. So they tick a lot of boxes, but one box that they didn't tick is that we didn't have a way to create internal heterogeneity within these condensates. We didn't have ways to arrange different molecules in different locations of these aggregates, which, as we said, it's really an important ingredient in engineering synthetic cells, because you want to be able to spatially organize different functions. So the question that I'm going to address now is, how do we establish internal heterogeneity within these DNA condensates? So there's a couple of different ways in which this can be achieved. And one of them that I'm going to focus today is by basically implementing molecular reaction networks within these condensates that sustain diffusion reaction processes. The way in which we can do this is by modifying our elementary building block, attaching a long bit of DNA, so about 40 nucleotides, that we call a binding site. And then we consider some other DNA, which is present in solution, and we call these patterning strands. So in this example, we have two patterning strands, a short one, which is labeled with a red fluorophore, and it binds to this short red domain here. And then a long patterning strand labeled with a green fluorophore, which binds to the whole of the binding site. What we're going to do is that we're going to make these condensates with the binding site present in all of the DNA nanostructures, but no patterning strands. And then we add the patterning strands in the outer solution. Now, because the condensate is porous, the patterning strands are going to diffuse inside this condensate. And they do so at different rates because they have different sizes. So the short strands, the red ones, will diffuse in first. And as they do, they will bind to the binding site. And therefore, you see this red front propagating inwards. But then if you wait a little bit longer, also the green strands will start diffusing in. They take longer because they're bigger, but eventually they do. And as they do, they actually displace, through a strand displacement reaction, the red strands that were initially attached to the binding sites. And the red strands get released back into solution. And therefore, you get this green front that progressively consumes the red patch. If you wait long enough here on the right-hand side, you can see that the condensate becomes all green because thermodynamically it is favorable for the binding sites to be occupied by the longer strands. But at these intermediate times, so here after 37 minutes, we have achieved what we wanted. So we have found a way to localize certain molecules only in the middle and certain molecules only on the outside. So create this internal heterogeneity. Now, these molecules are not very useful. They're just kind of fluorescent molecules. They don't do anything particularly interesting. But because this is DNA, you can easily functionalize it with useful molecules that carry functionalities. And so you can engineer functionality within your synthetic cells, as we're going to demonstrate in a bit. This is just going to be a video that shows the process which I have described. So you have the red strand going in first, and then you get that the center becomes red, followed by the green strand, and then everything eventually becomes green. We can, of course, do this with more than two strands if we want to have, say, more than two addressable regions within our synthetic cells. So in this example, we have a third strand, which has intermediate length between the red and the green, and is non-fluorescent. So this is going to generate a black region, looking black in confocal, between the red and the green. And you can see this in the video. So you have the red, then the non-fluorescent strand going in next, followed by the green strand. And after 12 minutes, in this particular example, we have created these three addressable regions within the synthetic cell, each enriched in one of these three unique strands. We can go with 5 strands, see if we can create 5 concentric rings. So here we have 3 fluorescent ones, 2 non-fluorescent ones. This video goes quite quickly and is a little bit mesmerizing as well. But if you look at the kind of still of the video after about 7 minutes and you stare at it for enough time, you can convince yourself that there are 5 concentric rings. So you have red, black, green, black, and then green. And you can also verify this with a fluorescence cross-section. And the maximum we could achieve, which isn't actually in the published paper, is 7 rings, which we could create with 9 strands. So here again, some of them are fluorescent, some of them are not. So the video here also goes quite fast, but after 30 minutes, again, if you stare at this long enough, you will notice that there are 7 concentric rings. And this is really the maximum we could do, because as you can see here, then the strands become quite similar in size, only different by a couple of base pairs, which makes the diffusion constant similar. And also the toehold for the strand displacement reactions become very, very short. So the strand displacement becomes slow as well. So all of this is not really useful if you let it equilibrate to the point in which all of the binding sites become occupied by the longer strands, because then you lose your pattern. But it's actually very easy to arrest these processes at any time you want by adding an excess of this stop strand, which has the same sequence as the binding site. You add it in solution, in excess, it captures all of the leftover patterning strands, arresting the process. And you can see this in this video. After about 16 minutes, you add the stop strand, and the patterns simply become arrested, and they remain frozen for a very, very long time, indicating that we can indeed kind of freeze these patterns, and they remain frozen simply because the patterning strands will not come off spontaneously. They are long enough that they remain attached. They can only come off if they are displaced by another patterning strand. So if you remove the patterning strands, then the patterns remain frozen. We can model these using reaction diffusion equations implemented in a spherical geometry. I'm not going to enter into the details of how this is done for reasons of time. These are the equations for a single strand diffusing into a condensate. You can have similar equations for 2 strands, 3 strands, as many strands as you want. You can use these reaction diffusion models to fit your experimental data. The way in which we like to plot this experimental data is by having the time on the vertical axis, and then the distance from the center of the condensate in the horizontal axis. And then this is the fluorescence intensity sampled radially in your condensate. So if you look at this diagram up here for the single strand that diffuses in, basically what you see here is the front progressing from the outside to the inside of the condensate. The model does a good job fitting this data, as you can see, for 1 strand, 2 strands, and 3 strands. And you can use this model to extract useful parameters like the diffusion constants of the strands, which follow trends that make sense based on the length of the strands, as well as the binding and strand displacement rates. So to conclude, I'd like to show you how we can use these reaction diffusion processes to achieve what we wanted, which is create a synthetic cell which has this kind of internal separation of functionalities. And what we wanted to build here is a very simplified synthetic cell, which has a nucleus in kind of big air quotes where RNA is made, surrounded by a cytoplasm with even bigger air quotes where the RNA is accumulated. And the way we did this is by localizing in the center of this synthetic cell with a variation of these reaction diffusion processes, a long bit of DNA, which is actually a template with a T7 promoter that codes for a broccoli aptamer. So the polymerase can go into the center and make some broccoli aptamer. So this broccoli aptamer has been extended to have this little handle. And then we have located in this outer shell a binding site for this broccoli aptamer. So the idea is that you do this patterning. So you put the template in the middle, the binding site on the outside, then you add your polymerase, which is this red thing. It will diffuse in, it has enough space to do so. It will make the RNA in the center. Then the RNA will become fluorescent, binding to the DFHBI, will diffuse outwards and then accumulate in this outer shell. We're going to see this happening in this video here. So here in cyan, we have the center, so the nucleus of our synthetic cell. And in orange, you're going to see the broccoli being made. And as you can see, it actually accumulates from the inside outwards because it's being made in this central cyan region. And you can see this in this sequence of confocal steels. So the orange again is the broccoli accumulating. And if you look at the bright field image of this object, it does actually grow in size. So it's just making RNA and accumulating RNA. And all of this work on the reaction diffusion processes was done by Adrian, who was a PhD student in the group who recently discussed this thesis.
Lorenzo, could you please wrap up in a few minutes?
Yes, this is my last slide. So this is just kind of some news, if you wish. I'm not going to have time to discuss this in detail, but basically you can achieve similar structures and even more complex structures by playing with phase separation of these amphiphilic DNA building blocks. And also you can obtain morphological responses to RNA production. And this is discussed in this preprint that we just posted on bioRxiv last week. And with this, I'm going to conclude by thanking all of the amazing collaborators involved in this research listed here, all of the group members who directly contributed, who are highlighted, and I've shown their pictures as well, some funding, and finally, I would like to thank you for your attention.
Thank you very much. Questions for Lorenzo?
Just one thing that I didn't quite get clear in the penultimate scientific slide. Was this a fluorescent broccoli aptamer, or was it? Did this involve any polymerase?
Yes, sorry, maybe I went a little bit fast there because I was because I was running out of time. I'm gonna it's a fluorescent broccoli aptamer which is made by T7 polymerase. So I can share my screen again. Share. So if we go here. No, here. So we have located in the center of this object a template for the broccoli aptamer, which has a double-stranded promoter for T7 polymerase. And so the polymerase will diffuse in and then make the broccoli aptamer, which will bind to the DFHBI, and then then we'll kind of diffuse outwards and accumulate in this outer shell. So it's made by polymerase. Yeah.
That's good. Thank you. Thanks.
Yep.
Nice talk, Lorenzo. I just noticed the timing of your RNA synthesis in this, and it went on for hours before you see actually the accumulation of the broccoli outside, right? How does that compare with actually the kinetics of the endogenous RNA? Is somehow the RNA synthesis slowed down because it's embedded in this, or can you use it somehow to infer kinetics of RNA synthesis using your strategy?
Yeah, that's a great question. I mean, it's kind of hard to predict, like to compare, because it's not very easy to figure out exactly how much template you have in your sample in these particular samples. But from some experiments that we have done, and they are in the SI of this paper, I think it's significantly slower compared to what you would have in the bulk. And I think this is just because this is a very complex environment. Probably the template is kind of twisted around this network of DNA, and the polymerase needs to make its way through. So I'm not surprised that it's lower, but it's significantly slower. Again, with the caveat that we don't know exactly how much of the template we have overall in these samples.
Okay, let's thank Lorenzo one more time.